Imagine for a second that you are tasked with creating a map.
Right.
But not just any map. You need to map every single star in the entire Milky Way galaxy.
Which is a massive job on its own.
Yeah, exactly. But there is a catch. You don't just need to know where each star is. You also need to know the exact chemical composition burning inside every single one of them.
That sounds completely impossible.
It does, right? Well, today, we are going to talk about a team of scientists who essentially accomplished just that.
But instead of mapping the galaxy, they mapped the mammalian brain.
Exactly. Welcome to today's deep dive. We are exploring a truly groundbreaking 2026 paper from the journal Neuron by Murakami and colleagues.
It is a phenomenal piece of work.
It really is. Our mission today for you listening is to understand a revolutionary piece of technology called MFISH3D and the artificial intelligence that powers it, which is known as ZenCell. Together, these tools allow researchers to look at a whole intact organ, like a mouse brain or a piece of human tissue map, millions of individual cells within it, and identify exactly what genes each and every cell is expressing.
It really is a remarkable leap forward for biology because, for over a century, our biological imaging has been mostly trapped in 2 dimensions.
Right, on those little glass slides.
Exactly. We would take a piece of tissue, slice it incredibly thin, place it on a glass slide, and look at it under a microscope.
Which is fine for basics, but
biological organisms don't exist as flat slices.
Yeah.
They operate in a fully intact 3-dimensional ecosystem. By finally moving from 2D slices to a massive 3D volume, we aren't just getting a prettier picture.
we're seeing how they actually live.
Yes, we are preserving the spatial relationships between the cells. And that spatial neighborhood is where the real biological communication actually happens.
Okay, so let's unpack this. Because the very first major hurdle this paper tackles is a highly physical, chemical one.
Chemistry is everything here.
Yeah. How do you look inside a thick, completely opaque piece of tissue, like an entire brain, and see the exact genetic code being expressed inside individual cells?
Right, because the old way of doing this, spatial transcriptomics, had a pretty glaring flaw when it came to thick tissues.
It just couldn't scale, right?
No, it lacked scalability entirely. If you tried to reconstruct a 3D volume by stacking hundreds of 2D slices together digitally, you inevitably ended up with gaps, tears, and distortions in the tissue.
Because you literally cut it with a blade.
Exactly. Or if you tried to stain a whole organ using traditional antibodies, it would take weeks for the chemicals to penetrate.
Wow.
And the results deep inside the tissue were incredibly unreliable. That is where FISH comes into play.
FISH.
Yes, FISH stands for fluorescence in situ hybridization.
I've always found the concept of FISH fascinating, but how does it actually work in practice, like step by step?
Think of it like sending a highly customized glowing puzzle piece into the brain. This puzzle piece is synthetically designed so that it only snaps together with one specific genetic sequence, a target messenger, RNA, or mRNA.
So it ignores everything else.
Right. If a cell is actively expressing that specific gene, the puzzle piece binds to the mRNA, and that particular cell lights up under a laser.
That sounds simple enough in theory.
In theory.
Yeah.
But getting those fragile puzzle pieces deep into a whole, fatty, intact brain without destroying the delicate mRNA inside the cells, that requires engineering a highly specific chemical environment.
So it's not just a matter of soaking the brain in a dye.
Not at all. The rigor of the chemistry is what makes MFIS3D possible. The very first step is getting rid of the fat in the tissue.
Because brains are incredibly fatty.
Exactly. Lipids scatter light. They have a different refractive index, which makes the tissue opaque, and they physically block our fluorescent probes from getting inside.
So they have to clear it out.
Yes, they perform delipidation. The researchers found that soaking the tissue in hot methanol at 50 degrees Celsius was the optimal way to strip the fat out.
That makes perfect sense if you think about washing a really greasy frying pan.
How so?
Well, if you use cold water, the grease just smears around. You need hot water and a strong solvent to strip the fat away.
That is a great analogy.
But here you have to be careful not to scrub so hard that you damage the delicate nonstick coating, which in this biological case is the fragile mRNA. I noticed they strictly limited this hot methanol bath to under 10 days.
Precisely. You are walking a biochemical tightrope to prevent the RNA from degrading. Once the fat is gone, you have to introduce those fluorescent puzzle pieces.
Right.
This happens in a liquid called the hybridization buffer. And instead of just guessing, they engineered a microscopic delivery system.
What's in it?
The buffer contains a very specific mix. It's 1 molar sodium chloride, 40% formamide, carefully balanced at a pH of 7, all kept at exactly 37 degrees Celsius.
That sounds like a very specific recipe. Why those exact ingredients? Let's break those down.
Every single component has a critical protective job. The formamide acts as a denaturant.
Meaning it unfolds things.
Yes. Alongside the gentle 37-degree heat, it essentially relaxes the tightly wound nucleic acids, helping the fluorescent probes penetrate deep into the dense tissue.
Okay, that makes sense.
But there is a danger there. If you soak tissue in a warm liquid, it wants to absorb water. It hyperhydrates, swells up, and literally falls apart.
Oh, wow. So you're trying to open the tissue up without letting it dissolve into mush.
Exactly. That is why they use a massive salt concentration, the 1 molar sodium chloride.
Ah, to pull the water back?
Yes. It acts as an osmotic stabilizer, protecting the tissue's structural integrity.
Right.
And keeping the pH exactly at 7 provides the optimal environment for the probes to bind strictly to their targets.
Rather than just sticking randomly everywhere and creating background noise.
Precisely. You want a high signal-to-noise ratio.
But just one microscopic glowing puzzle piece isn't bright enough to see through a whole organ. You need to amplify that light.
Yeah.
The paper mentions they use something called hybridization chain reaction, or HCR, to boost the signal. But how do you stop that amplification from going out of control? I mean, you'd think adding more probes would give you a stronger, better signal.
You would think so, but biology is rarely that straightforward. To get the amplification ingredients deep into the tissue, they used a polymer called PEG 20,000 to help pull the chemicals through the dense cellular matrix.
Okay.
But here's the brilliant part. They discovered that lowering the primary probe concentration and running the reaction at 37 degrees Celsius was the secret to clean data.
Wait, using less probe gave them a better result.
It did because of the blood vessel.
The blood vessels.
Yeah. Blood vessels are notoriously sticky in these types of biological assays. They love to trap fluorescent probes, creating massive false positive signals that look like glowing genes, but are actually just trapped dye.
Oh, that would ruin the whole map.
Entirely. By optimizing the thermodynamics at 37 degrees and using fewer probes, they ensured the amplification only happened exactly where the target mRNA lived. It was a massive technical victory.
That is incredibly clever. But they still have one major physical barrier, making the tissue invisible so the microscope can see the glowing cells inside.
The clearing process.
Right. They use a clearing solvent called BAB, but apparently ran into a wall with the standard chemical buffers.
Yes, they did. The standard buffer used to wash the tissue before clearing contained sodium citrate.
Okay.
But when sodium citrate mixes with methanol during the clearing process, a chemical reaction occurs that turns the whole solution cloudy and opaque.
Which completely defeats the purpose of trying to make the organ transparent.
Exactly. It's like a fog rolling in. So the researchers substituted the standard ingredients with lithium chloride and malleate.
Why lithium malleate?
Lithium malleate stays perfectly dissolved and invisible in methanol. It solves the precipitation issue and grants perfect optical transparency.
So now we have a transparent brain. But I read something in this paper that sounded almost destructive.
The bleaching.
Yeah. They mixed hydrogen peroxide into the clearing solvent and exposed the whole brain to intense light. Isn't pouring bleach on a brain going to completely destroy the delicate RNA you're trying to map?
It sounds totally counterintuitive, but it's a technique called H2O2 assisted photobleaching.
How does that not destroy the sample?
Well, a natural brain actually has a built-in glow under a microscope because of natural pigments in the cells. It's called autofluorescence.
Oh, so the brain is glowing on its own.
Yes, and that background glow drowns out the delicate signal of our fluorescent probes. The hydrogen peroxide, when activated by light, specifically breaks down those glowing pigments without harming the robust nucleic acids.
That's amazing.
They systematically destroyed almost 68% of the brain's natural background glow.
I love that visual. It is like turning off every single streetlight in a massive, sprawling city just so you can finally look down and clearly see the fireflies.
That is exactly what it's like.
So before we move on to how they actually read this map, let's take a breath and look at what this team has engineered.
A quick recap.
Yeah, let's do a quick recap for you listening. They've essentially solved the physical problem of 3D mapping. They use hot methanol at 50 degrees to strip the fat, like degreasing a pan.
Under 10 days.
Right. They use a high-salt, temperature-controlled buffer to safely drive the probes deep inside without turning the brain to mush. They swap in lithium malleate to make the whole organ perfectly clear like glass.
Avoiding that cloudy sodium citrate.
Exactly. And they use hydrogen peroxide to turn off the city's streetlights, leaving a pitch-black background for the glowing genes. It's an absolute masterclass in chemistry.
It really sets the stage.
Which makes me wonder, for you listening right now, if you could use this chemical recipe to make any solid object in your everyday life completely transparent just to see its inner workings, what would you choose?
It is a fun thought experiment.
Yeah.
But of course, in the lab, this raises an entirely new, massive challenge.
Right, the data.
Exactly. The chemistry allows us to see our genetic fireflies, but how do we make sense of millions of them? The paper promises a 10-plex map.
Meaning 10 different genes.
Yes, that means identifying 10 entirely different genes operating simultaneously in the exact same brain.
And here is the math problem with that. The standard high-end microscopes they use only have 4 laser colors available.
The 488, 561, 640, and 785 nanometer lasers.
Right. If you only have 4 colors of light, how on earth do you map 10 different genes at the same time?
You don't do it at the same time. You have to do it in sequential layers.
Oh, I see.
You stain the tissue with your first batch of probes, let's say 4 different genes. You put the whole transparent organ under the microscope and image the entire 3D volume.
Okay, that's round 1.
Then you take the brain out, use chemicals and light to wash out and destroy those specific fluorescent tags, and you restain the exact same brain with the next batch of probes.
Wow.
You image it again, and you repeat the process. They did this for 4 full rounds.
But hold on. You are taking a squishy, jelly-like brain, moving it in and out of microscopes and chemical baths for weeks on end.
Yes.
How do you ever line up the images perfectly again? If the physical brain shifts, squishes, or stretches by even a fraction of a millimeter, your map of millions of cells is completely ruined when you try to digitally stack the layers later.
That physical distortion is exactly what ruins most 3D spatial biology. But the researchers solved it with a brilliant counterstain.
What did they use?
In every single round of staining, no matter what other targeted genes they were looking for, they dedicated one laser color to stain for a constant.
A constant?
Ribosomal RNA, or rRNA.
Oh, I see. It acts like a permanent GPS anchor.
Exactly. Ribosomes are everywhere in the cytoplasm of a cell. So the RNA stain creates a persistent, highly detailed structural map of every single cell's body.
So it's like a landmark.
Yes. When they feed these massive images into their software, and we were talking about 150 gigabytes of data per scan, the computer doesn't look at the whole organ.
It looks at the landmarks.
Right. It looks at the RNA pattern from round 1, round 2, and so on, and uses those physical cell bodies to digitally snap the images together.
The algorithm they use to align these colossal data sets is called Big Stitcher. And it does this in stages, right? Because 150 gigabytes is massive.
Yes, it works from the macro down to the micro. First, it performs a global affine transformation.
What does that mean visually?
Think of this as taking a digital picture printed on a rubber sheet and just roughly rotating and stretching the whole sheet until the outer borders line up.
Just getting it in the ballpark.
Right. But because the brain tissue squishes locally during handling, they then move to a global dense deformable registration.
Okay.
This corrects the uneven stretching across the whole tissue. Finally, it uses chunkwise dense deformable registration, correcting local microscopic distortions down to a resolution of about 2.6 by 2.6 by 4 micrometers.
That is incredibly tiny.
And at incredible scale, you are aligning single individual cells perfectly across weeks of separate imaging sessions.
Here's where it gets really interesting, because once you have this flawlessly aligned image showing 10 different genes across an entire intact brain, you hit a human limitation.
A massive one.
A grad student cannot sit at a computer and manually click on and count millions of individual 3D cells. It would take multiple lifetimes. You need an automated system.
Enter ZenCell.
Yes, ZenCell.
ZenCell is the artificial intelligence they custom built for this exact pipeline. The architecture relies on what is called a vision transformer, or VIT.
How is that different from older AI?
Older AI models, like convolutional neural networks, essentially scan an image pixel by pixel through a tiny sliding window. They look at a tiny piece, then move over, then look at another piece.
Like looking through a keyhole.
Exactly. A vision transformer does something entirely different. It looks at distinct patches of an image and mathematically understands how all those patches relate to each other simultaneously, regardless of how far apart they are on the screen.
But how does the AI know what a cell looks like if it's never seen one? I read that they use a technique called masked autoencoder, or MAE, pre-training.
Yes. Yeah.
And what blew my mind is that they trained this AI initially without any human labels telling it what a cell is. How does it learn without an answer key?
That is the beauty of MAE pre-training. They feed the AI a biological image, but they digitally hide or mask random patches of it.
Like a jigsaw puzzle with missing pieces.
Right. The AI then has to guess what the missing pieces look like based on the surrounding context. To do this, they give it a small target area, a region of interest, of 256 by 256 pixels.
The ROI.
Yes, but crucially, they also feed it a massive context area of 1280 by 1280 pixels.
Okay, let me make sure I'm wrapping my head around this. It's essentially like teaching an AI to understand the overarching concept of a forest, that massive context area, before you ever ask it to outline the specific leaves on a single pine tree, the region of interest.
That is a perfect analogy.
By forcing it to learn the context first, the AI figures out the fundamental structure of biology on its own.
Because it grasps the forest, it becomes incredibly robust at dealing with the messy, varied backgrounds, the uneven lighting, and the dense crowding you inevitably get in 3D biological imaging.
It knows what belongs and what doesn't.
Exactly. Once it understands that broad context, they fine-tune it. They give it a small number of human-labeled 2D slices.
To give it the actual vocabulary.
Right. The AI learns from those human examples, and then it does something remarkable. It generates its own 3D pseudo-labels.
Pseudo-label.
It extrapolates from the 2D data, creates a draft 3D map of the cells, and the researchers just proofread its work.
And the results were staggering. In their benchmark tests, ZenCell significantly outperformed heavy-hitting, widely used existing algorithms like CellPose and Stardust when it came to 3D segmentation. It even beat SwinUnetter.
It wasn't even close in some of those complex 3D environments.
So let's summarize this data pipeline really quick. We beat the 4-color laser limit by washing and restaining the tissue in rounds.
Using RNA as our GPS.
Right, ensuring those rounds line up perfectly for the big Stitcher algorithm. And finally, we unleash the ZenCell AI, which learns the forest before the trees, to automatically segment millions of 3D cells without human bias.
A fully automated mapping system.
It really forces a perspective shift. For you listening, does trusting in artificial intelligence to automatically map millions of brain cells make you nervous about leaving humans out of the loop? Or does it make you incredibly excited about the sheer speed at which we can now discover things?
That speed of discovery brings us to the most important part of this paper.
The actual biology.
Yes. Establishing a groundbreaking chemical protocol and a cutting-edge AI is an incredible engineering feat. But the true test of any tool is what it actually reveals about biology.
Right. If you build a telescope, you have to point it at the sky.
Exactly. Murakami and the team didn't just build this tool. They deployed MFISH3D across several distinct biological cases to prove its real-world utility.
And this is where the practical application really comes into focus.
Yeah.
Let's look at their first case study, the drugged mouse brain.
A classic pharmacological test.
They injected mice with a drug called MK801. Now, for some context, this is an NMDA receptor antagonist. It basically blocks a specific type of signal in the brain and is frequently used by researchers to model the symptoms of schizophrenia in lab animals.
A very standard model.
Right. They wanted to see exactly which cells in the entire brain reacted to this drug. To do this, they tracked a gene called FOS.
FOS.
Yeah. When a neuron fires and becomes highly active, it turns on the FOS gene. So seeing FOS is basically seeing a glowing sign that says, I just fired.
But tracking FOS alone only tells you where activity happened, not who was active.
Just that a light went on.
Right. Because of the multiplexing capability of MFISH3D, they simultaneously mapped 3 different subclasses of inhibitory neurons, marked by the genes PVALB, SATES, and VIP.
So they are looking at the exact network.
Exactly. They wanted to know exactly which type of inhibitory neuron was being disrupted by the schizophrenia drug.
And the results were shockingly specific. They found that the PVALB neurons barely reacted to the drug at all.
Almost silent.
Yeah. But the SACE neurons, their activation went up 3.6-fold. And the VIP neurons massively spiked, showing a 4.2-fold increase in density in the cortex.
Think about why that matters for a moment. If you are a pharmaceutical company trying to develop a new psychiatric medication with fewer side effects, you don't want a drug that blasts the whole brain.
You want a sniper rifle, not a shotgun.
Precisely. You want a drug that highly targets the specific SMACE-VIP networks without touching the PVALB networks. Furthermore, because they mapped entire intact brains, ZenCell caught inter-individual variability.
Meaning differences between mice?
Yes. It noticed that the response in a specific area, the retrosplenial cortex, varied slightly from mouse to mouse. You completely miss that nuance if you were just blending up a piece of tissue and running a standard genetic sequence.
It's a total game changer for drug development. But they didn't stop at pharmacology. In case 2, they looked at developmental biology. Yeah. They took a mouse embryo at embryonic day 13.5 and mapped a region of the developing brain called the diencephalon. They wanted to trace the lineage of cells expressing a gene called CALB1 versus GAD1.
GAD1 being the marker for GABAergic neurons.
Right, the neurons that act as the brain's braking system.
Tracing developmental lineages in 3D space is notoriously difficult because embryos are constantly shifting and growing. It's a very dynamic environment.
But MFISH3D clearly revealed that the CALB1 expressing cells have a completely different developmental origin from the GAD1 neurons in that specific area.
Which rewrites how we understand brain development.
And to prove the chemistry isn't just limited to the brain, they imaged an adult mouse kidney in the same study.
Right. The kidney is incredibly dense, non-neural tissue. It's a tough environment for imaging.
But they successfully mapped the intricate 3D structures of the nephrons, the microscopic filtering units that clean our blood.
They captured the whole architecture.
They could actually see the gene pod axle strictly confined to the glomeruli, the tiny filter tufts, while SLC7A13 wound through the proximal tubules and CALB1 in the distal tubule.
Understanding that precise 3D architecture is vital for understanding kidney disease.
Which brings us to case 3, a fascinating detour into evolutionary biology, the squid brain.
Specifically, the big fin reef squid.
Right. Cephalopod brains are essentially alien compared to mammalian brains. Their evolutionary path diverged from ours hundreds of millions of years ago.
And we have very few tools designed to study them properly.
But with MFI-SH3D, they didn't need specialized squid tools. They just custom designed the genetic puzzle pieces and went right in. They mapped a gene called EAT, which marks glial cells.
Glial cells being the vital support cells of the nervous system.
Yes, and they discovered something amazing, a highly regular, tiled sublayer structure of glial cells right in the inner granular layer of the squid's optic lobe.
We're literally charting unexplored evolutionary terrain at that point.
It's incredible. But as profound as mapping a squid brain is, Case 4 represents perhaps the most immediate medical significance, the human brain.
This is the ultimate test.
Applying any kind of spatial biology to human postmortem tissue is notoriously difficult. The tissue is often archived, it's been frozen for years.
And the biochemical quality varies wildly depending on how the person died and how the tissue was stored.
Exactly. They acquired samples from the NIH Neurobiobank, specifically looking at Brodmann Area 9 and the cingulate cortex, Brodmann Area 24.
These are regions heavily involved in complex thought, emotion, and memory.
And to my amazement, the protocol worked flawlessly. On this old, archived, frozen human medical tissue, they successfully mapped the SENT, MP, and PLP1 genes across all 6 distinct layers of the human cerebral cortex.
If we connect this to the bigger picture, it proves this technology is not just a delicate novelty for perfectly prepped lab mice.
Right, it's not just an academic exercise.
Exactly. It is a robust, viable technique for studying the human brain right now. Whether it is tracking the mechanistic response of a psychiatric drug, untangling the embryological origins of a kidney, exploring the evolutionary marvel of an alien-like visual system in a squid, or probing human cortical architecture for disease markers.
This technology seamlessly transitions across the entire domain of biology.
It really does.
It's a lot to take in, so let's synthesize these 4 biological leaps for the audience. We've seen how tracking precise neuronal reactions to drugs can revolutionize pharmacology. We've seen how mapping embryonic lineages and 3D kidney filters can rewrite developmental biology.
We've charted the alien architecture of a squid's optic lobe.
And if proven we can pull high-definition maps out of archived human medical tissue, it begs a massive question for the future. Of all these applications, drug testing, embryonic development, exploring uncharted evolutionary biology, or charting the human cortex, which field do you think will fundamentally change medicine first?
Synthesizing everything we've explored today, the journey we are on is quite profound.
We've come a long way from the glass slides.
Yes. We have finally stepped away from the flat glass slides that constrained biology for a century. We are now entering a highly detailed, chemically preserved, 3-dimensional ecosystem.
We are taking entire organs, making them invisible.
Selectively illuminating their deepest genetic conversations.
And using advanced artificial intelligence like ZenCell to catalog millions of data points without human limitation.
It's a new era of biology.
We've given you, the listener, the shortcut to the absolute bleeding edge of neuromapping today. You now understand exactly how scientists are merging hot methanol baths, clever osmotic balances, and vision transformer AI to chart the brain.
The foundational tools of the next decade.
But before we sign off, I want to leave you with one final provocative thought, something to mull over that builds on everything we've discussed today.
Indeed. If we can now map every cell's genetic state, its precise type, and its exact 3D location in an intact brain, consider what happens when we apply MFISH3D to an entire human brain across different stages of aging.
Or tracking a devastating disease like Alzheimer's.
Exactly.
Think about the implications of that. Could we map the precise, microscopic chemical moment a memory network begins to fail? Before a single memory is actually lost by the patient, could we use this exact technology to see the very first protective neurons flickering out in a 3D physical space?
Finding the spark before the fire.
If we can see the fireflies going dark before the forest burns, could we finally learn how to stop the fire? It is a wild, hopeful thought. Keep questioning the world around you, keep exploring, and thank you so much for joining us on this deep dive into the 3D cellular universe.